Hello, my name is Nicole Lima, and this is my Week 3 Sensory Physiology portfolio assignment. I'll be presenting on the anatomy and physiology of the retina, but I also wanted to talk about the process by which the eyes and the brain communicate in order to transduce the light that we see into visual images. So I wanted to start off with a brief overview of the process of vision, which can be separated into 3 steps. So the first is that the lens focuses light onto the retina. The second is a process called phototransduction, in which photoreceptors in the retina transduce the light into electrical signals. And the third process is signal processing, in which the electrical signals are converted into the visual images that we see. So starting off with this first step, the lens is what is responsible in the eye for focusing light onto the retina, and it does this by a process called accommodation. So the lens is attached to the ciliary muscles by these zonule fibers, and the ciliary muscles contract and relax to alter the curvature of the lens. So when objects are far away, the ciliary muscles are relaxed and the lens assumes a flatter shape. But when objects are closer to the eye, the ciliary muscles contract, causing the lens to become more rounded, which increases the angle of refraction of light that comes into the eye. And this is all done so that the light can be focused onto the retina and provide us a clear image. So moving on, we have some anatomy of the retina. So the retina contains 5 different types of neurons. So the first type is the photoreceptors here, which are the cones here in purple and rods in yellow. So the cones are responsible for color vision and high acuity vision, and the rods are responsible for low light vision. As you can see here, the rods outnumber the cones about 20-1, and this is the case everywhere in the eye except for the fovea, which we'll come to a little later. Next, we have the horizontal cells, which are just cells that mediate lateral inhibition, which enhances the location and contrast of stimuli that is processed by the eye. Next, we have bipolar cells, which synapse between the photoreceptors and the ganglion cells. We also have amacrine cells, which modulate the flow of information from the bipolar cells to the ganglion cells. And then finally, we have the ganglion cells here in blue, and their axons are what make up the optic nerve that go to the eye, to the brain. And then finally, we also have the pigment epithelium, and it's this dark color because of a pigment called melanin, and its responsibility is to absorb excess light in the eye so that our vision is not distorted. And then over here, we just have another view of the retina. So we have the fovea and then the macula, and this is the center of the visual field, and it's also the area with the highest acuity vision. This is because there are just cones here in this area, no rods, and there are no blood vessels or anything else in the way. So you get really clear, sharp vision here. And then we also have the optic disc here, which is where the optic nerve leaves the eye and goes to the brain. This is also known as our blind spot because there are no photoreceptors here. So next we will come to phototransduction, which is the process by which photoreceptors convert light into electrical signals. So here we just have a close-up of a rod cell, and rods and cones contain visual pigments. In rods, that pigment is called rhodopsin, which is just made up of 2 separate components. So first is a protein called opsin, which is this purple dumbbell-shaped structure here, and then retinal, which is this red structure. Retinal is a vitamin A derivative that allows us to absorb light, and together they are called rhodopsin. And then another thing to know is that there are 3 different types of channels within a rod. So CNG channels, which stand for cyclic nucleotide gated channels, and these allow positive cations to come into the cell. And then there are also potassium leak channels, as well as voltage gated calcium channels, which are not shown here. So when the cell is in the dark in this panel here, rhodopsin is inactive, and cGMP levels in the cell are high. This causes the CNG channels to be open, which allows for an influx of calcium and sodium ions, and The potassium leak channels are also open, which allows potassium to leak out of the cell. Since the influx of positive ions is greater than the efflux of positive ions, the cell is depolarized to about negative 40 millivolts, which causes the calcium channels to open, and this causes a release of glutamate onto the bipolar cells. And something to keep in mind is that when the cell is in the dark, this is a tonic release or continuous release of glutamate. The next panel here is when the cell is exposed to light. So when the cell is exposed to light, rhodopsin becomes bleached, which just means that retinal changes conformation and is released from opsin. This activates a cascade in which cGMP is decreased in the cell. CNG channels close, which prevent positive ions from coming into the cell, but the potassium leak channels are still open, and this causes the cell to hyperpolarize to about negative 70 millivolts. And this is a graded response, so in bright light, no glutamate will be released because the calcium channels will never open. But in dim light, some calcium channels will open and a little bit of neurotransmitter will be released. And then finally here in this last panel we have the recovery phase, which is when the cell goes back into a dark environment. So retinal gets converted back to its inactive form and then recombines with opsin to form rhodopsin. And this is actually the reason why the time that it takes for retinal to go back to opsin is why our eyes take sometimes a little while to adjust between light and dark environments. So next here we have signal processing, which is when electrical signals are transferred into the visual images that we see. So in this image here, this is a great example of convergence. So as you get further and further from the photoreceptors, you have fewer and fewer cells synapsing. And even though there are about 5 or 6 rods here, in a real cell, there are about 15 to 45 photoreceptors that may synapse onto a single ganglion cell by the end. And so something that occurs here is that ganglion cells receive information through specific areas of the retina called visual fields, which you can see here. So they have a center and a surround, which is this gray part. And then, depending on where the light is hitting this visual field, certain bipolar cells are either activated or inhibited. And there are 2 types of bipolar cells. There are on-bipolar cells and off-bipolar cells. So in on-bipolar cells, when they are exposed to light, the cell is excited, and when they are in the dark, the cell is inhibited. And the opposite is true for off-bipolar cells. So in the light, the bipolar cell is inhibited, but in the dark, the bipolar cell is excited. And then based on where the light is also hitting the visual field, the ganglion cell responds in a different way as well. So ganglion cells respond most strongly when there is a strong contrast between the surround and the center of the visual field. So if there's a strong contrast, then there will either be a lot of action potentials that are sent, or there will be none. But if the contrast is weak between the surround and the center, then there will be an intermediate response by the ganglion cell, which you can see in this chart here. So for an on-center, off-surround visual field, if bright light goes onto the center, the ganglion cell is excited. But for an off-center, on-surround visual field and bright light goes onto the center, the ganglion cell is inhibited. And then the opposite is true for if the bright light goes onto the surround. If the field is on-center and off-surround, the ganglion cell is inhibited. But if the field is off-center and on-surround, the ganglion cell is excited. And then kind of the same thing when there is diffuse light or a weak contrast between the the center and the surround, the ganglion cells respond weakly. And here is just a study aid that I made because I had trouble myself with the concept kind of the on-bipolar cells, the off-bipolar cells, and the on-center and off-surround ganglion cells. So just to follow this flow chart, we have a photoreceptor. If it responds in the dark, we have an inactive form of rhodopsin. This leads to high cGMP, open CNG channels, and open potassium leak channels. This depolarizes the cell to negative 40 millivolts. Calcium channels open, and then you have a tonic release of glutamate. If this is an on-bipolar cell, glutamate binds something called mGlu-R6, and the bipolar cell is inhibited. But if it's an off-bipolar cell, glutamate binds and opens some ion channels, and the bipolar cell is excited. On the other hand, if the photoreceptor responds to light, rhodopsin gets bleached, there is low cGMP in the cell, CNG channels are closed, and potassium leak channels remain open. This causes the cell to hyperpolarize to negative 70 millivolts, calcium channels remain closed, and no glutamate is released. If we're talking about an on-bipolar cell, mGlu-R6 is not activated by the glutamate and the bipolar cell is excited. But if we're talking about an off-bipolar cell, no glutamate is released, so the ion channels remain closed and the bipolar cell is inhibited. And then here on this side, for the visual field, for the ganglion cells, if it's an on-center, off-surround visual field and you have bright light on the center, the ganglion cell is excited. But if you have the same visual field and the bright light is on the surround, the ganglion cell is inhibited. And then if you have an off-center, on-surround visual field, bright light on the surround portion of the visual field causes the ganglion cell to be excited, but bright light onto the center causes it to be inhibited. So I hope that is a helpful study aid for others as well. And then finally, we just have what happens past the retina. So once the signal goes into the optic nerve, it crosses the optic chiasm where some of the nerve fibers cross over. Then it goes to the lateral geniculate nucleus, or the body, in the thalamus. And then finally it goes to the visual cortex in the brain. And so something to notice is that everything from the left visual field is processed in the right visual cortex, and then everything in the right visual field is processed in the left visual cortex. And that is the end of my presentation. I hope you came away with a better understanding of the anatomy and physiology of the retina, as well as how light is transduced into visual images, and I will see you next time.